Hello everyone, welcome back for more human physiology. So we are nearing the end of our unit on the endocrine system, so one of the last major glands for us to discuss is the pancreas and its hormones. So let's waste no further time and get right after it. Oh, excuse me. So the pancreas is interesting in that its endocrine activity is not its only major function. So the pancreas also functions in an exocrine role. If we want to get a feel for its location, it's in the abdominopelvic cavity, so it's below the level of the diaphragm. So your thoracic cavity is above the diaphragm where your lungs and heart are located. Everything else that we would talk about is below there, so the pancreas is below. It is situated in this little J-shaped loop of the intestine, and then it is also located inferior and situated posterior to the stomach. So if you're looking at the stomach, the pancreas should be right behind it and a little bit below. As I was mentioning, most of the cells of the pancreas do not produce hormones. Most of the cells of the pancreas are exocrine in nature, meaning their products that they secrete are going to an external surface rather than into the blood. So most of what these exocrine cells are going to do is synthesize digestive enzymes and deliver them through the pancreatic duct, as you can see here in green, to the intestine so that we can further break down our food so that we can absorb nutrients into the bloodstream, as we have discussed before. So if you want to focus on the endocrine nature of the pancreas, then you have to look for these little patches of cells called islets of Langerhans, or just pancreatic islets. So these islets contain several different types of cells called alpha cells, beta cells, delta cells, and F cells, which are each going to secrete their own particular type of hormone. So if you look at this picture, the yellow cells are, again, what we say are about 99% of the cells in the pancreas. They are exocrine cells called acinar cells, and they are going to synthesize and secrete digestive enzymes. At the center of this, you can see one of these pancreatic islets, and you, of course, can see some infiltrating capillaries, which is important, of course, because once we secrete these hormones that we're about to talk about, they've got to find their way into the blood supply. So, as I was saying, there are four different types of cells here. Alpha cells, which secrete glucagon, beta cells which secrete insulin, delta cells which secrete somatostatin, the same somatostatin that is also secreted by the hypothalamus, although it does not act as a hormone in this case. And then finally, the F cells secrete a hormone called pancreatic polypeptide, which really only has effects on regulating the functions of the digestive system. So our focus is going to be just on the alpha and the beta cells, so just insulin and just glucagon. So by now, what you are probably familiar with is that both insulin and glucagon are involved in regulating your blood glucose levels. So all for the sake of homeostasis, we try to keep the levels of glucose in the blood within a normal range, usually somewhere between 70 and 100 milligrams per deciliter. But what's the purpose of this? Why do we want to keep blood glucose regulated? What's the point? Who cares if it gets too high, right? Who cares if it gets too low? So glucose is one of those monosaccharides that we talked about in Chapter 2, and it is a very common starting material for cellular respiration. A lot of cells in the body, pretty much all the cells in the body, are capable of using glucose for making ATP, provided that oxygen is available, right? So most of the cells in the body don't have to use glucose. All cells can use glucose, but most cells can actually use other things like proteins and lipids to make energy. But here's why we have to have glucose around. The brain is a very picky eater. It's like a 2-year-old that only wants to eat macaroni and cheese and chicken nuggets, right? So you can't really get the cells of the brain to do cellular respiration with anything other than glucose. You can't get it to feed on lipids. You can't get it to feed on proteins. So this is why it is a problem if you end up with hypoglycemia, meaning blood glucose levels that are persistently below the normal range, below 70 milligrams per deciliter, because the cells of the brain will therefore not be able to meet their energy demands if they don't have glucose to feed on. So the downside of having too much glucose, which is called hyperglycemia, is a lot more complicated, and we could spend half an hour talking about that, but I'll save you that discussion. But we will know that Hyperglycemia is very heavily linked to diabetes mellitus, which of course we will discuss much later. So insulin and glucagon, before we really get into their regulation, we introduced them earlier in this chapter as antagonistic hormones. What that means, again, to reiterate, both hormones, both antagonistic hormones, will have control over the exact same parameter, which is your blood glucose. They will produce changes to that parameter in opposing directions, meaning glucose will drive the blood glucose levels down, and glucagon will drive them up. And as a consequence, the two hormones are very unlikely to be found in the blood at the same time, as their actions would essentially cancel each other out. The whole thing would be a zero-sum game, and it would be counterproductive. So the way to think about this, as always, is insulin and glucagon are going to maintain homeostasis. We've got this normal range, as indicated by this green part of this gauge right here, the 70 to 110 milligrams per deciliter blood glucose. We're trying to keep things balanced. We don't want it to get too low, which is hypoglycemia. We don't want it to get too high, which is hyperglycemia. So another way to kind of think of this balance is kind of like a seesaw. So when a seesaw is balanced, the two ends are perfectly level with each other. So when blood glucose is balanced like this, there should not be a need for either one of these hormones, neither insulin nor glucagon. But of course, as you go about your day, your blood glucose levels are definitely going to fluctuate a lot. So take one example. Say when you eat a meal. So when you ate your breakfast this morning, whether it was a pastry or a donut or a muffin or something like that, it contains carbohydrates. So when you digest those carbohydrates and break them down into individual glucose molecules, you absorb that through the intestines, like what we talked about in chapter 3, right? So that is going to increase your blood glucose levels above, pretty close to the top of the normal range, if not above. So this is going to shift our balance here towards hyperglycemia. And in this case, the beta cells in the pancreas are going to be the ones that sense this. So this is a simple endocrine reflex. None of this that we are discussing here with the pancreas has anything to do with the hypothalamus or the anterior pituitary. So the pancreas is acting alone here. It senses, the beta cells sense the high blood glucose, and they secrete insulin. The idea here is that insulin will reduce the levels of glucose back down into the normal range. So here's how it works. So we know that our trigger, our stimulus, is the high blood glucose. The beta cells of the pancreas are acting as both the sensor and the control center here, and our output signal is the insulin itself. The insulin, being a hormone, gets into the bloodstream, circulates all throughout the body, and it goes looking for its effector cells. In this case, skeletal muscle, adipose tissue, and to a lesser extent, liver tissue as well. So keep in mind, as always, I feel like I've been hammering this into you at this point, but just in case it hasn't kind of sunk in, effector cells, target cells, they are going to be the ones that have receptors for this particular hormone. So only skeletal muscle, adipose tissue, and liver tissue are going to have insulin receptors. And once insulin binds to these receptors, it is going to activate a signal transduction mechanism that will bring the blood glucose down. But how does that happen? Well, let's take a look at it. So the underlying issue is that, as we've discussed back in chapter 2, glucose is a very polar molecule, so it's not going to be able to just directly diffuse across cell membranes, right? We talked about that in chapter 3 as well. So that high amount of glucose compared to a low amount of glucose inside the cell creates a concentration gradient, but because glucose can't cross the membrane without some help, nothing's going to happen, at least not until insulin comes along. So as we point out here, glucose, if it's going to cross the membrane, it's going to need a carrier to let it across. We have these carriers in liver, skeletal muscle, and adipose tissue cells. These are called GLUT4 channels. But the problem is that without insulin, they are not in the plasma membrane. They're actually stuck inside the cell in these secretory vesicles, and they are therefore non-functional and cannot let glucose in. So when insulin binds to the insulin receptor, which is a tyrosine kinase type receptor, this is going to activate a signal transduction cascade that will cause these GLUT4 channels, which are embedded in secretory vesicles. It will cause those channels to fuse with the plasma membrane, causing the insertion of these channels into the actual cell membrane, and now these channels are all ready and willing to allow glucose to come into the cell, which effectively will reduce the amount of glucose that was in the blood previously because it's all inside the cell now. So with all this glucose that's now inside the cell, we can either use this glucose right away for cell respiration, or if we want to save it for later, we can store it up as glycogen in skeletal muscle and liver tissue, or we can convert it to fat in adipose tissue. So if we want to conclude what we've talked about here with insulin, this is the pancreatic beta cells which secrete insulin when they sense that blood glucose levels are getting high. So since insulin causes those target cells of the liver and the muscle and the adipose tissue to take glucose from the blood, this removes glucose from the blood and will cause the blood glucose to get back into the normal range there. So how do we keep an increase in blood glucose well balanced after we eat something? We do it with insulin. So insulin production, typical negative feedback, should stop once we get levels back to normal. There's no need to have that insulin in the blood unless blood glucose levels are very high. So what about the opposite scenario? What if it's been quite a long time since you've eaten? And for someone like me that can't tolerate being hungry, this is about the biggest tragedy that I'm going to talk about in this chapter, having been a long time since you've eaten. Nobody likes that. So it's the same sort of thing. If it's been a long time since you've eaten, your blood glucose levels are going to start to decrease as your cells use what little glucose is there for cellular respiration. Again, this will shift the balance, and this time the alpha cells of the pancreas are going to respond by secreting the other hormone, being glucagon. This time, our intent is not to reduce blood glucose, but to raise it back up into the normal range. So the mechanism this time, it's going to be quite similar. The alpha cells of the pancreas will act as both sensor and control center, sensing the low blood glucose levels, secreting glucagon as our output signal. Glucagon will get into the bloodstream the exact same way insulin did, and the effectors are even going to be the same as well: skeletal muscle, liver, and adipose tissue. So all three of these tissues not only have insulin receptors, but they've got glucagon receptors as well. And once glucagon does its thing, which we'll talk about on the next slide, the blood glucose levels should start to rise. So the glucagon receptor is actually a G-protein coupled receptor. So the mechanism behind how it works is going to be totally different. So when glucagon binds to this G-protein coupled receptor, the idea here is that any stored up glycogen in the liver or skeletal muscle will be broken down through a process called glycogenolysis, in which enzymes cleave the glycosidic bonds in stored up glycogen to free up glucose, and then glucose can exit the cell via facilitated diffusion to get into the extracellular fluid so that we can increase the amount of glucose in the blood. So let's actually talk about glycogen, because we've mentioned it a few times, but we haven't really had a whole lot to say about it specifically. So glycogen is a very long polymer that is made up of many glucose molecules bound together. So we can find glycogen in both skeletal muscle and liver tissue, mainly in the liver tissue. You store up about 5 pounds of glycogen in your liver. So when glucagon binds to receptors on our target cells, we stimulate glycogenolysis by activating some of those enzymes that chew up the glycogen. So this should free up individual glucose molecules, and we can export them out to get them into the blood to increase our blood glucose levels. So here is a very interesting thing to think about. Why would it be better to store glucose as a polymer rather than just storing up a whole bunch of individual glucose molecules inside the cell? So it says think back to chapter 5, but that is actually mislabeled. It means think back to chapter 3 and some of our discussions there. So again, why would it be better to store glucose as a polymer rather than just storing up a whole bunch of individual glucose molecules inside the cell? So I'm not going to give away the answer to this. I would like you to think about it and definitely post to the discussion board and have a conversation with your classmates about this, and we'll see if someone can come up with the correct answer here. So to wrap up our discussion on glucagon, this time it is pancreatic alpha cells which secrete glucagon when they sense that blood glucose levels are getting low. So again, the action of glucagon will balance things out right here by liberating glucose so that we can secrete it into the bloodstream and get those blood glucose levels to rise. Negative feedback should work the same way. Once the stimulus is gone, once blood glucose is back to normal, glucagon production should go back to normal as well. So let's talk a little bit about pathology here. So obviously we want to talk about diabetes. The most common cause of hyperglycemia is going to be diabetes mellitus. So what diabetes is a pathological deficiency in insulin signaling. So you, I'm sure, are aware that this comes in two varieties, type 1 and type 2. Type 1 is a genetic issue and an autoimmune issue. Type 2 is something that you acquire later in life because of lifestyle choices and things of that nature. So the way that this is supposed to work, as we saw before, is that when blood glucose levels get high, the beta cells of the pancreas sense this, secrete insulin, insulin binds to target cells, which take up glucose from the blood, and that reduces your blood glucose. Type 1 and type 2 diabetes represent a failure of this mechanism, but they represent two different failures, although the end result is essentially the same, a failure to bring blood glucose levels down. Type 1 diabetes represents a failure to make insulin to begin with. Basically, the way this works, as I said, this is an autoimmune disease. Basically, the body mounts an immune response that kills off the beta cells early in childhood so that because there are no beta cells, a person with type 1 diabetes cannot make their own insulin. So treatment for them is fairly simple. You can just give them insulin injections. The treatment is simple, although that's not to minimize the inconvenience and suffering of someone who has type 1 diabetes. It's certainly harrowing, but theoretically, conceptually, the treatment is rather simple. Type 2 diabetes does not represent a failure to make insulin. People with type 2 diabetes make plenty of insulin, if not more insulin than the average person. Their problem is at the receptor level. Basically, insulin has been in the blood for so long and so commonly that they have developed an insensitivity to insulin, meaning that their receptors no longer bind to and react to insulin the way that they should, meaning that the body tries to make extra insulin to compensate for this. So really their problem is that they make insulin, but they just can't really respond to it well, so their blood glucose stays high either way. Type 2 diabetes treatment is a little bit more complicated. You can give them more insulin, although that's not going to be a cure for it. There are other drugs out there like metformin, which are given to type 2 diabetes patients in an effort to make the cells more sensitive to insulin. So just to kind of finish this out, this doesn't really have anything to do with the pancreas, but before we start talking about endocrine pathologies in our next video, just a list of a couple of other organs with endocrine functions. I should have mentioned this in the first video, so this may just be a little bit of repeat information. The heart makes a hormone called atrial natriuretic peptide, which helps to signal to the kidneys to reduce sodium reabsorption. The GI tract makes three different enzymes, gastrin, CCK, and ghrelin, which either help aid in digestion or signal to you that you've eaten enough and you are full. The kidneys, we've mentioned, make two hormones, renin, which is a part of that renin-angiotensin-aldosterone system that we talked about before in regulating blood pressure and fluid balance. Erythropoietin is the signal for us to make more red blood cells when systemic oxygen levels get very low. Adipose tissue makes a hormone called leptin, which, similar to ghrelin, helps us to feel full. and the liver makes a enzyme slash hormone called angiotensinogen, which is going to, again, like renin, be part of the angiotensin-renin-aldosterone system. All right, so that's going to do it for this video. We are almost done with the very long Chapter 17, so join us next time for one more video, and we will talk about endocrine pathologies. Thanks for your attention, and I will see you next time. Bye-bye.